All right. Good afternoon, everybody. Hope you guys are doing well. We're finally getting a break from all the rain we've had the last few days. So finally get to see some sun and not have to worry about getting wet outside, which is very, very nice. Hopefully that keeps up. Um, I guess a couple announcements. I got all of the exams graded earlier this morning and then got the exam grades put into Canvas before class started today. So if you didn't see that or get the notification or whatever else, you can go into your grade book. You should see the column showing the percentage score that you got from the lecture exam, the lab column showing the percentage score that you got from the muscles lab exam, and then your exam 2 average, which is the actual score contributing to your overall points, which is taking both percentages, adding them together, and dividing by 2, right? Getting the average. So for everyone who has taken both exams, your grades for that should already be up and available for you. So check that out. I will bring your exams with me to all of our future labs. So if at any point you want to see your exam and what you missed points on or argue with me about anything, I'm happy to do that like usual. So for today's class, oh, that froze my screen. For today's class, Zoom. Uh-oh, you guys have disappeared on my end. Oh, there we go. Okay, looks like Zoom froze for a second. For today's class, as I was saying, I want to start off going over the action potentials questions with my key. So this little worksheet that some of you guys worked on at the end of class on Tuesday, so we can talk about action potentials. And then we'll jump into the chapter 13 content, which we basically started in lab yesterday with the structure, the anatomy of the spinal cord, and we'll expand on some of that stuff. I'll re-review that structure stuff. All that. Okay. So if you want to reference what you guys worked on last week or on Tuesday, I'm gonna send both documents into the chat. If you want to have that pulled up while we talk about it, you have that available for you. Can't get to this third tab. Move this, there we go. Here's group twos. Okay, and then here's my key. So in this first question, you guys were given a graph showing the membrane potential over time of what it would typically look like as an action potential moves across the neurolemma on that portion, or the axolemma on this portion of the axon. So we have our resting membrane potential signaled by A here at -70 millivolts. You then see that membrane potential take a huge spike up into the positives and then start to come back down with C, even going beyond our resting membrane potential, dipping into -80 before it gets back to the same charge that it started with. And so these are the major steps of an action potential. All action potentials, so this change in membrane potential along the axon, are going to look like this. There is no change. They are all or nothing. So no matter how strong the stimulus or how frequent the stimulus, this is the same pattern of change of potential that is going to occur. It always looks like this. It doesn't get, there is no larger spike or greater dip. It always ends up like this for each action potential specifically. And so in part A, I ask you guys to label B, C, and D as either depolarization, hyperpolarization, or repolarization. And so depolarization, if we deconstruct that word, we're assuming that this is starting off as being polar, which it is. It has a strong negative charge, so the opposite side of the membrane will have a positive charge in comparison, giving us our two poles. And so by bringing that charge closer to zero, that is depolarization. So in B, this large jump from negative up to positive to match the opposite side, we are shortening the distance between those poles, shortening the difference in charge between those poles. So this is depolarization. C, as that charge is going back, dipping back down into the negatives, that is repolarization, right? Going back to having that large difference in charge. And then this portion D, where we've gone beyond the resting membrane potential, dipping even farther into -80. Hyper means above or more than, and so we're going beyond the original charge. So this is hyperpolarization. So depolarization, removing that difference in charge. Repolarization is getting it back to that difference, and then hyperpolarization here, going beyond the original charge. And so this is all caused by the movement of ions across the membrane. And in order for these ions to move in large numbers the way they want to, the way they would be forced to do by their electrochemical gradients, we need to have some ion channels opening. And so these are all looking at our voltage-gated ion channels. So the charge of the membrane stimulates each of these differences, except for, I guess, our original sodium channel opening, which would typically be a chemically gated, ligand-gated channel. So anyways, for depolarization, this initial change in potential, the sodium channel is going to be open. So sodium is flooding into the cell, bringing all of that positive charge with it. So bringing a bunch of positivity to the internal environment of the cell, thus making the charge more positive as it rushes up here. When it reaches about plus 30 millivolts, the sodium channel is going to close and the potassium channels are going to open. And so now we have those positive ions flooding in the opposite direction. So during this depolarization, sodium is flooding in, then the sodium channels close, the potassium channels open, and now potassium is going to flood out of the cell. So that's where we see this repolarization of the charge becoming more negative, because potassium is also positively charged. So it's taking positivity out of the cell, making that charge more negative. And potassium channels are slow to close. And so once we have reached our resting membrane potential again, that is going to start signaling our potassium channels to start closing, but they don't close immediately. And so we get this short period of time where we are hyperpolarized or have an even larger positive charge, negative charge than we started with. And that's where we get that little dip down into -80. Eventually, all those potassium channels will close, and then the sodium-potassium pump will restore resting potential back to -70. So some sort of stimulus opens a sodium channel, allowing those positive sodium ions to flood into the cell. That depolarizes the membrane. At plus 30, the sodium channels close, potassium channels open, and now that positivity is leaving the cell. So we get repolarization. At -70, the potassium channels will now close, so will start to close, so now both channels should be closed or closing, but potassium channels are slower to close, so we get this short period of hyperpolarization until they finally are all closed, and the sodium-potassium pump will restore the original potential by pumping out 3 sodiums and pumping in 2 potassiums. So removing some of that positivity at a slower rate. Okay. Did you guys have any different answers here? Or is there anything to discuss that might seem off or seems confusing or doesn't match up logically? Okay. Again, all action potentials will look like this. They follow the same exact pattern. Action potentials are the same every time. They are all or nothing. If we reach that stimulus to start an action potential, it is always going to start. So if we reach that -60 millivolts, the rest of this is always going to occur in this pattern. So in question 2, we have essentially these same steps that you saw in the graph being written out. And I asked you to order these. There's 7 different events, so ordering them 1 through 7, 1 being what happens first and 7 being what happens last. And so I've basically made a different copy of this same table to put them all in chronological order the way they would be to make it easier to talk about. But these are the numbers that should line up on the table that you were working on. So anyone who is working on that and wants to check their numbers, it should be 3, 1, 4, 7, 2, 5, and 6. So now we'll talk about it in the order that things happen. So again, at rest, the voltage on the membranes of our axons is -70 millivolts. Or I guess of the neuron in total is -70 millivolts. In this case, an excitatory postsynaptic potential is going to stimulate the depolarization at our axon hillock, which is going to start an action potential. So this excitatory signal depolarized the membrane to threshold. -60 is the threshold that causes an action potential to go. And it did this by opening sodium-gated channels, which are going to cause other voltage-gated sodium channels to open. So now that those channels are open, sodium is going to quickly flood into the cell, depolarizing the charge, so making it much more positive, up until around +30 millivolts. At this charge, the sodium channels are going to close and the potassium channels are going to open. So that's what was next. Upon reaching the peak of this action potential, voltage-gated sodium channels are inactivated and voltage-gated potassium channels are activated. So now no more sodium is coming in, or at least not a substantial amount of sodium is coming in, and a whole bunch of potassium is flooding out of the cell. So the charge is going to start becoming negative again. These channels are really slow to close. So even once they pass that -70 millivolts, they are going to continue to send potassium out of the cell until eventually they do completely close. But that's what causes that hyperpolarization. And then a little bit of repolarization again as, or wait, sorry, I just realized how strange that fits into the middle here. I think this is actually supposed to be 5 and 6. So just a second here. That probably was supposed to look more like that. So let me see what I did here. 5, 6. Okay, sorry, mixed those up. Because the event here at 4 showed that potassium channels are opened. This is describing the change in potential as a result of that. So as potassium floods out, we get repolarization as the charge is becoming negative again. Until it reaches -70, those potassium channels are starting to close again, but they're slow. So we get some extra potassium leaving the cell, which causes hyperpolarization. And lastly, all those channels are finally closed and the resting membrane potential will gradually be restored thanks to that sodium-potassium pump. And again, I know I've said this many times, videos are going to be the most helpful thing with getting all of this stuff down, getting to see it happen step by step with both events essentially being talked about. Seeing it happen while it's being described is really, really helpful in learning this stuff, getting an understanding of it. Part 3 here, I asked you to identify a few statements as either true or false and to give a quick explanation if any of them are false. So this stuff is all going to heavily align with kind of the summary table of action potentials and then our comparison of graded potentials versus action potentials, understanding the difference is kind of what I'm looking at here, or trying to help you guys learn with this. So part A states that voltage-gated sodium channels are quick to open and slow to close, while potassium channels are slow to open and quick to close. Both types of channels are very quick to open. They respond to voltage. So as soon as the threshold, the voltage is reached, that channel is going to open quickly. Sodium channels are quick to close, but potassium channels are slow. So the sodium channels are basically all close very, very quickly as we reach that +30 threshold. But potassium takes a little bit longer, and that's why we get that hyperpolarization. And hyperpolarization helps prevent excessive stimulus, excessive action potentials being sent again and again. It kind of causes what we call a refractory period. There is a small period of time in between each action potential. No matter how much stimulus is coming in, there must be some sort of pause in between while the cell gets back to its resting potential, and then it will respond to that sodium flooding in again, essentially. All right, so B says before a second action potential can be generated, the concentration of sodium and potassium on either side must be fully restored. That is true. That's basically what I was just describing, that refractory period. So we need to get the cell back to that -70 millivolts before another action potential can be stimulated. C says the strength of an action potential is represented by the amplitude of the wave. A stronger stimulus will generate an action potential with a higher peak than a weaker stimulus. So this is not true. This is actually describing graded potentials. So again, I was saying earlier, all action potentials will follow this same pattern, reaching the same charges every time. And there is no stronger action potential or weaker action potential. If the axon hillock of the axon reaches that negative 60 millivolt threshold, an action potential will start no matter what. It just has to reach that, and then it's all or nothing. If it doesn't get there, it won't start. If it does get there, it will always look like this. They are always the same. So this is false. Action potentials are all or nothing. All stimuli that exceed threshold will produce the same response. However, graded potentials, which are what cause action potentials, can be stronger or weaker, and they are the sum of their stimuli. So our graded potentials are what are starting at the axon hillock. I thought I had a different picture to talk about, but this is good for that. In order for an action potential to travel down the axon, the neuron must be stimulated at the axon hillock by a graded potential. So we can have multiple neurons synapsing with one neuron at the same spot, or just have multiple signals coming from the same neuron over and over again at that axon hillock. But it is the combination of each of those signals, whether they are excitatory or inhibitory, the combination of those changes is what is going to determine whether or not an action potential starts. So if the combination of those signals reaches that negative 60 threshold, the action potential will always propagate down the entire axon, and it will look the same. But graded potentials can be different. They can be inhibitory, they can be excitatory. It is the sum of those signals that starts the actual action potential. Okay, is everybody following so far? Is there anything from these that you would like to discuss? Okay, and our last three here. D says that action potentials travel in a non-decremental manner down the axon, with the voltage constantly being regenerated along the way, unlike graded potentials, which quickly diminish over a short distance. Again, that goes back to that all or nothing thing. If an action potential is started, it is going to do that and have the same change in charge along the entire axon, having that same effect. Graded potentials, however, do quickly diminish over short distances. Okay. E is talking about our two different forms of propagation of action potentials. This is really just talking about the difference between a myelinated or unmyelinated axon. So a myelinated axon is going to have these fatty myelinations insulating these portions, these internode portions of the axon. And what this does is it traps any of the ions that have moved into the cell at some other segment. So if an action potential started here and a bunch of sodium flooded into the cell, that sodium is going to stay inside the cell as it diffuses down the axon. So we get a depolarization all the way along here until it reaches the node. Then it will depolarize that node. We have also our potassium channels opening, etc. So really we have those events just repeating at each node of Ranvier. So this myelination allows the action potential to kind of skip some steps. It doesn't have to do all of the movement along the entire axolemma. It can skip from one node to the next node to the next node. And that is called saltatory propagation. And that is much, much faster than having to continuously depolarize, repolarize, hyperpolarize the entire axolemma, the entire membrane of the axon here. So myelination, providing this insulation, just makes those things happen faster. So saltatory propagation is our fastest movement. Contiguous propagation is much slower. That is describing basically doing all of that depolarization along the entire membrane. So contiguous propagation requires the opening and closing of voltage-gated ion channels to depolarize the entire axolemma, continuing the action potential. With saltatory propagation, we get to maintain that membrane potential across the entire portion of the membrane that is insulated, And depolarization only needs to occur at the nodes around BA. So much less that needs to occur to allow that action potential to keep happening or to continue to travel. And then lastly, action potentials originate in the axon hillock and travel down the axon all the way to the synaptic terminals. That is true, right? Graded potentials happen at that axon hillock where the body meets the axon. If it reaches threshold, that action potential will go all the way to the end of the axon, reaching the synaptic terminals. So yeah, that was the little activity. So again, I'll open up one final time for any questions or concerns. I hope for those of you that did work through this and use this practice, I hope that was helpful in kind of putting these things in order and identifying some of the bigger differences between graded and action potentials and some of the main ideas of how action potentials work. So yeah, thanks for participating. And so for the rest of the day, we can talk about our chapter 13 content. So yesterday in lab, we introduced the structure, the anatomy of the spinal cord, right? You guys were making basically this image here. We zoomed in to this part with a lot of this. Okay, this is better. This with all of the meninges surrounding it, all the connective tissue. Okay. So this is what the inside, this is a transverse section of your spinal cord. So this is the organization of all the neurons that are traveling from or to your brain, and then from or to your entire body in order for us to perceive sensations and then to send out our responses. And so I'll go slide by slide for today, basically talking about it as we haven't talked about it before. There. So the topics discussed in this chapter, or the topics to focus on in this chapter, are recognizing the gross anatomy of the spinal cord, again, what you did in lab, recognizing the spinal meninges, which are all of those connective tissue layers, the dura mater, the arachnoid matter, the pia mater, recognizing the organization of gray matter in the spinal cord, white matter in the spinal cord, anatomy of a spinal nerve, and then stuff that we're not going to talk about directly in lecture, like I'll touch on it, on what they are, but the nerve plexi, which are just large organizations or how we group various spinal cords based on what areas of the body they innervate. So which areas of the body they are responding to sensations from and sending motor signals to. So we have four main plexi or organizations: our cervical, brachial, lumbar, and sacral. So the cervical basically serving the neck and shoulder region, the brachial plexus serving the pectoral and upper limbs, the lumbar plexus serving the pelvic and lower limbs, and then or lower trunk, and then sacral plexus helping with those lower limbs and lower trunk as well. We introduce dermatomes, and then lastly talk about the reflex arc, or a few different examples of reflex arcs. All right. So essentially, our peripheral nervous system has two main types of nerves. And again, peripheral nervous system is just anything leaving the central nervous system. So these nerves that extend beyond the brain and spinal cord. And so the nerves that leave directly from the brain are called the cranial nerves. And the nerves that leave from the spinal cord to go throughout the rest of the body are our spinal nerves. So we have those two major types: cranial, directly from the brain; spinal, coming from the spinal cord. So in this chapter, we're looking at mainly spinal cord nerves and its structure. And so we name our spinal nerves, and each nerve here, so C1, is not one single nerve, it is one pair of nerves. There is a nerve going out the left and the right side of this. So this is really referring to those groups, those pairings. And so we have our cervical spinal nerves, but you may notice already, we have 8 cervical spinal nerves here. We have C1 through C8, but we only have 7 cervical vertebrae. And so each of these nerves is extending out superior to the vertebrae that correlates with it. So C1 is coming out of the spinal cord above the C1 vertebrae, above the atlas. And so that stands true for C1 through C7. Right? 1 is above 1, 2 is above 2, 3 is above 3, etc. But then 8 is actually above our T1 vertebra, our first thoracic vertebra. And so for the rest of these spinal nerves, they are leaving inferior to the related vertebra. So T1 is actually leaving the spinal cord below or inferior to the T1 vertebra, and that continues all the way down into our sacral, and then lastly our coccygeal nerve, which I guess doesn't actually come out of the coccyx. But anyway, so they do not directly correlate exactly with the regions of the vertebral column that we've talked about, but those patterns do generally give you a really good idea. Again, cervical nerves coming out superior to the related vertebra, thoracic, lumbar, and sacral nerves coming out inferior to the related vertebra. I guess I should have been using this slide here. So 31 pairs of spinal nerves total. Again, these are pairs. It's one nerve going left, one nerve going right at each of these places. And then we have a couple other structures here to talk about. A couple enlargements. So the cervical enlargement and the lumbar enlargement. These are large groupings of nerves leading towards specific areas, such as our upper limbs and lower limbs. So the cervical enlargement has a lot of nerves leaving at the same time because they are going to go innervate all of our upper limbs. The lumbar enlargement has a lot of nerves leaving at the same time because they are all going out to our lower limbs. So those regions can be visually seen by that enlargement, that big grouping of them leaving. So the spinal cord itself technically ends, I guess, at the conus medullaris. I think what this means as far as where it ends is it is no longer going to have this same structure that we're seeing here. We're going to get a lot more exteriors, and the meninges are a bit more spread out or look a bit different. And so the general structure that we're talking about of how the spinal cord looks is going to stay pretty consistent in its organization all the way until that conus medullaris is around L1. Beyond that, we have a ligament. So the dura mater of the spinal cord will become continuous with connective tissue that ends up creating our filum terminale ligaments, which is within our coccygeal ligaments. Okay, so these are extensions of those nerves within that connective tissue. And we get this structure that looks like a horse's tail because we have all these nerves no longer being fully condensed into that same structure that we were seeing before, called the cauda equina, which means horse tail in Latin. So that's that structure where these nerves are starting to spread out, where the neurons that were previously all bundled together into the spinal cord are now much more spread. So some terminology. Again, I know we introduced this yesterday, but going to reintroduce it again. So terminology to look out for because you're going to see it show up in multiple places. We have gray matter versus white matter. We have dorsal and posterior being used to refer to the same location, and then ventral and anterior being used to refer to the same direction or location. And then our central canal visible in the center there. So gray matter is mostly made up of unmyelinated axons and neuron bodies. So James and I discussed at the end of lab yesterday that in the neuron bodies, you have those Nissl bodies, and the floating endoplasmic reticulum in our neurons. You remember this image, these Nissl bodies. These are rough ER that are not continuous with the nuclear envelope like they would be in most of our other cells. They are just floating around in the cytoplasm of the neuron. And those Nissl bodies tend to give the body of the cell of the neuron a kind of gray coloration. And so the gray color of gray matter comes from those cell bodies. And we have those Nissl bodies absorbing and reflecting some light, and that makes it look gray. And so these regions of gray matter are mostly neural bodies and unmyelinated axons. The white matter, which is making up most of the exterior here, is mostly or is all myelinated axons with some unmyelinated axons. So these are all axons. So any information receiving is going to happen in the gray matter. Right? Axons or neurons receive information from the dendrites, which are usually quite close to, if not directly connected to the body. And so most of the information receiving is going to happen in the gray matter, and then information leaving is going through the white matter, through those mostly myelinated axons. And then I introduced that these terms for direction, dorsal and posterior, as well as ventral and anterior, are referring to the same direction. We do have these two different terms because in different organisms, they are referring to different stuff. Dorsal means to the back. Or I guess, what is a better way to say that? Like towards the vertebral column, really. Dorsal means towards the vertebral column, whereas posterior means backwards, away from where the head would be facing. So in a human, dorsal and posterior are the same thing. Right? Our head faces this way, our vertebral column is behind that, and so our dorsal and posterior are in the same direction. But if you imagine something that would be on all fours, or like a dog or a dolphin, their vertebral column is pointing towards the sky, so that would be superior. But their head, the opposite of their head, posterior, would be towards their butt. Yeah, so it gets a little confusing with that. But we have two terms meaning the same general direction in humans. Dorsal or posterior mean the same thing. Ventral and anterior for humans means the same thing. So even though we're going to see that terminology show up in two different ways, like our anterior median vision and our ventral root, the direction is generally the same. So dorsal and posterior meaning towards the back, ventral anterior meaning towards the front. And we generally get the same organization of where information or how information is traveling into or out of our spinal cord based on those directions. So most of our sensory information that is going to need to travel to the brain to get some sort of response, that sensory information is going to leave posteriorly or dorsally. Motor information, which is typically going to be coming from the brain to go towards our muscles or towards the cells or towards a gland to initiate some response, those are mainly leaving anteriorly towards the front of the spinal cord. So we get some organization of that. And then we have this central canal in the very center, this tiny little hole running all the way down, just full of cerebrospinal fluid. So surrounding that spinal cord, again, these are just neurons. This is the organization of neurons. These are neuron cells running up and down in the center of your vertebral column and that vertebral foramen or the vertebral foramina, multiple. These are just neurons. And the nerves are made up of neurons. You can kind of see that we have connective tissue surrounding the nerves here. And we do have connective tissue surrounding the actual spinal cord as well. And so these layers of connective tissue are called the spinal meninges. These are our connective tissue layers. And we have three layers, just with different organizations of the types of protein filaments throughout this. So the outermost is the dura mater. Dura, you can think of durable. This is the toughest fibrous layer of the three. So the dura mater is the outermost. Deep to that would be the arachnoid mater. It is much more web-like, so less thick organization of the protein filaments and much more spreading, making almost a web-looking structure, thus arachnoid mater, referring to spiders. And then the deepest of our connective tissues is the pia mater. So that is what would be directly surrounding the spinal cord itself. And then we have some space in between, and each of the spaces has their own names. So outside of the dura mater, we have our epidural space, epi meaning outside. So outside of the dura, our epidural space. And the epidural space is mostly fat. It's there to insulate and protect your spinal cord. So you have this layer of adipose tissue in between your spinal cord and the vertebra that is surrounding it. So that if your spinal cord is shaking around, it's not bumping into bone and being scratched and damaged. So that epidural space helps with protecting it in the anterior to posterior direction. Then below the dura mater, where's the label for it? Over here, yeah. No. Dura mater, arachnoid mater. Okay. Sorry, below the arachnoid mater, we have our subarachnoid space. Sub meaning below, so the subarachnoid space. And that is in between the arachnoid mater and the pia mater. So those two main spaces. What else to talk about here? Denticulate ligaments. So I told you that the epidural space here with that adipose tissue is protecting our spinal cord in the anterior to posterior direction. And then we have this fibrous connective tissue making up a ligament, which is deep to the dura mater, which is protecting it from that side to side, that lateral movement, trying to somewhat keep it in place and protect it from bumping into bone or coming out of place when it's moving laterally. So when we were looking, for those of you that did the dissections of the spinal cord yesterday in lab, the things that were sticking out of the spinal cord before you cut anything, those were definitely the nerves. And you would have had to cut into the dura mater and start removing that layer of connective tissue to see the denticulate ligaments. They do not extend beyond the dura mater. It's basically the connection of the pia mater to the dura mater. But yeah, they run in the same direction. They're just very much smaller and deep into that dura mater. So once you cut open that first fibrous outer layer and started to pull it apart, those little web-like things that you saw touching it, the thicker ones there were the denticulate ligaments on the side. That's good for here. Just to look at a real one, I'm trying to see if there was anything specific to point out here. Wait. Okay, are we doing okay so far? I know this is reading vocabulary. Okay. Okay, so now looking closer at the organization of the neurons and the different types of matter in the spinal cord itself. So back to neurons. Okay. The gray matter in the center of your spinal cord makes this butterfly-looking shape with our two big wings coming out from the center. And each of these horns, literally called horns, and they have their names based on their directionality. So the posterior large horn here is our posterior gray horn. This tinier little bump is our lateral gray horn. And then we have a much larger one on the anterior that we refer to as the anterior gray horn. And each of these contains portions of the neuron that are responding to different types of information, or groups of neurons rather, that respond to different types of information. So we have our somatic sensory neurons, so responding to information that we are from our environment, from our external environments. We have somatic sensory neurons in the gray horn, a little bit of our visceral sensory neurons in that posterior gray horn as well. So mostly somatic sensory information, a little bit of autonomic sensory information found in that posterior gray horn. Most of this lateral gray horn is dedicated to that autonomic motor info, so sending back signals. So the somatic, or the sensory info here would be traveling upwards to the brain to send that signal, tell the brain what we are perceiving, and then the response, the motor signals are coming back down in this anterior area. So for the autonomic system, you would find those neurons in the lateral gray horn. And then for our somatic system, or for our central and peripheral nervous systems, those signals are going to be traveling down the anterior gray horn. So cognitive motor control, things that we can think about in our brain, and we have some sort of cognitive control over how we are responding. That info is traveling in this anterior gray horn. All the automated stuff, the internal environment that's constantly being regulated, checked on, and responded to, that is mainly being sensed and responded to through the lateral gray horn with some overlap into the posterior. And then most of our sensory information in general is coming in through the posterior gray horn. And again, I've referred to this a couple of times. Most of our sensory information is going to be some sort of ascending pathway, taking info from the environment or from our internal body and sending it up to the brain, since the brain is our control center that is going to react to that perception. So those pathways are ascending, traveling up the spinal cord, and then most of our motor control, the response, is going to be descending, traveling back out. And they follow the same basic organization. So motor information will leave through our ventral roots, or I guess, never mind, that would be incorrect to say. No, that is true. I was thinking about the groupings of things. So most of our motor information is leaving through ventral roots to then go to the parts of the body that are going to respond to it. And most of our sensory information is going to enter through these dorsal roots. These ganglions, these bulbous looking things on our roots, these are just where you would find the cell bodies of the neurons that are a part of that root. So the cell bodies tend to group up together in the same way that we have most of our cell bodies in our gray matter here. Those cell bodies group together, making this bulbous looking structure that we call a ganglion. So that's why we seem to have more mass in that space. Okay. Be sure that's all the important info there. Just another vocabulary term, the commissure. So for our anterior white commissure or anterior gray commissure, posterior gray commissure, commissure is just referring to where axons cross the spinal cord. So sending a signal across the midline of our body instead. Okay. So lastly, for this first part of chapter 13 here, looking at how our nerves are organized. So that was all spinal cord, looking at how the neurons in the actual spinal cord traveling in our vertebral column, how those are organized. But once those neurons leave and become spinal nerves, they are now extending out into the periphery, part of the peripheral nervous system. They are organized very similarly to how we saw muscle cells being organized or muscle tissue being organized. So what's happening inside the cell is quite different, but the organization of the tissue looks very similar. We have bundles within bundles within bundles. So we have an epineurium, which is the connective tissue completely surrounding the outer portion of the nerve. So this is what essentially the dura mater will become when it extends into the peripheral nerve. So it was dura mater when it was in the spinal cord, and that becomes continuous with the peripheral nerve and becomes the epineurium, the outer neural connective tissue. Within these nerves, we have bundles of axons called fascicles. So each fascicle has its own layer of connective tissue, our perineurium. So epi meaning outer, peri meaning between. So this is our middle layer of connective tissue. And those are bundles of multiple axons traveling through it. And then each of those axons has a connective tissue outside of it called the endoneurium. They may have even more layers, like if there are Schwann cells there providing myelination, right? That is fat, or mostly fat. But that endoneurium itself is that connective tissue. So endoneurium around each axon, perineurium around these fascicles, which are bundles of axons, and then the epineurium surrounding the entire nerve, which has multiple fascicles traveling within it. So each nerve is going to have sensory information and motor information traveling through it. We have sensory information coming towards the spinal cord through that nerve, motor information leaving the spinal cord through that nerve. And so they have neurons pointed basically in opposite directions to send both types of signals in or out. See that in an actual electron microscopy image here, with blood vessels traveling in between. Got that blood supply going. See all those axons sticking out here. Okay. So before I move on from this, again, I recognize most of this was vocabulary and trying to give some context to the vocabulary, but do you guys have any questions? Things that came up when you were reading I could address about the organization and structure of this study. Okay. Okay. So now we get into a bit of the physiology. I know that was heavy anatomy. We can talk about some physiology. So for these next 13 slides or so, I'm not going to talk about everything because I would mainly be reading names of nerves and what part of the body they innervate. Um, they're I'm working on finalizing the study guide so I can share with you the actual list of nerves that you should be aware of. It's just going to be some of the major ones. Hopefully not too long of a list of nerves, but there are some that are important for lots of things. Let's see, so we can see a lot of them listed here. Some of the major ones you will likely be asked to learn are the radial nerve, the ulnar nerve, and the median nerve. Um, the ulnar nerve is the one that causes that funny bone reaction because it only has like a little layer of your skin and fat protecting it from being bumped. The median nerve controls a lot of your wrist and finger movements. That's what's connected to carpal tunnel syndrome. Let's see, ones of the lower area. You'll likely be asked to learn the femoral, the obturator, and the sciatic nerve. Right, so it's not going to be the full list of all the nerves that you're going to see pop up on these slides as you go through, but you should certainly be aware of each of the plexi and the general areas that they serve. So each plexi is referring to groups of nerves that are responsible for the same relative areas. So we have our cervical plexus, brachial plexus, lumbar plexus, and sacral plexus to be aware of and what they innervate. Um, all of the thoracic nerves in between, so T2 to T11, they're not part of any of these plexi lists. They mainly innervate the trunk, whereas the rest of these tend to extend a bit beyond the trunk. So yeah, these are mainly just reference, putting all the figures together for you guys. I will release the list of uh, which nerves to respond or to know before next week starts. I have a lot on my plate that I want to get done this weekend, and getting those study guides finished is part of that. Something else to briefly talk about here is dermatomes. So dermatomes are describing the bilateral, so both sides of the spinal cord that are monitored by a single pair of spinal nerves. So you can see from this image the entire region of the skin, sorry, specific to the skin surface. So skin surface regions that are monitored by one pair of nerves. You can see C4 mainly responsible for sensations along the upper back and then across the middle. Okay. So these lines are bilateral, they cover both sides of the body. Mainly being aware of what a dermatome is. You may be asked on a quiz what portion of the body would correspond with things, but I honestly don't know. I'm not sure how they even put together a good question to test you on or to assess dermatomes, or if that um, necessarily matters. But knowing what a dermatome is, I guess, is the important thing here. Okay. And then lastly, some of the fun physiology, reflexes. So we have some automated responses to things that can be cognitively controlled, can be inhibited or excited by putting significant thought into it. But we have evolved to naturally respond to these things, even though they are somatic. They're not supposed to be, they're not visceral, they're not part of our internal environment. So these are natural reactions to the external environment without needing to travel all the way to the brain to get that input back. We have some interconnection within our spinal column that is going to cause this reflex. So again, a reflex is somatic information and response that is kind of automated, that does not require the brain to be involved. And each of our reflexes is going to follow the same 5 basic steps. And so altogether, from sensation to reaction, that is our reflex arc. And it does make kind of an arc. So if you have a reflex to like feeling something that is burning hot with your finger, that sensory information travels all the way down the nerve to your spinal cord, across an interneuron, and all the way back out to your arm to tell you to flex away. and not touch that again. So a reflex arc is going to follow these same 5 basic steps. So you have your receptor, which is responding to whatever stimulus that is, stretch or damage, which could be from heat or directly being cut. And that's going to activate a receptor, or that stimulus activates the receptor neuron. The receptor neuron sends a signal down our sensory neuron. And so anything that is sensory is going to be afferent, think affect. So to affect something with an A is to be the verb of an affect. You are affecting something. To effect something with an E, that is the response, that is the noun. So our sensory neuron is sending the effect, sending the sensation to the spinal cord. In the spinal cord, there will be some control, some processor, which is going to be an interneuron. So an interneuron is what is connecting the sensory neuron to the motor neuron in this reflex. So inter meaning between. So the sensation comes into the spinal cord, that stimulates the interneuron sends that signal back to the motor neuron, and the motor neuron tells the muscles what to do. So we received the sensation, we have sent that sensation to the spinal cord. This processor is going to transmit the response that should be sent to the motor neuron, and then the motor neuron is the efferent signal, the effector, the response, which is going to tell our target cell how to respond, whether that's a muscle or a gland telling it to secrete a hormone. So our effector is that target cell, what is carrying out the response. Again, this is for most of our examples here, this is going to be contraction of a muscle. So the effector is the muscle that is contracting. But we do have the same reflex kind of thing going on with hormone secretion and glands responding to those signals. And so the interneurons, or I guess maybe it's better to show an image like this first. So let's talk about one example of this reflex arc. So the most simplistic version is going to be a monosynaptic reflex. So we have basically one single synapse or one single interneuron then going to cause one target cell or one grouping of target cells to respond. So in this case, we had the stimulus of feeling pain, pressure on the palm, feeling that sensation of damage on the palm. The sensory neuron sent that signal through the dorsal root into the spinal cord. Again, most sensation is coming in through the dorsal root. That then stimulates this interneuron, which connects the sensory neuron to the motor neuron. So this interneuron to stimulate a motor neuron. And that is going to cause our effector, our muscle cells here, to contract and get your palm away from that pain. And this happens really, really fast. And the faster it can happen, the better, right? Because if you don't have that sensation, that realization that there is this nail sticking into your palm, the longer it takes for you to get that, the more weight you put, the more damage that it's caused, and the more repair you have to give. It's biologically expensive, right, to respond slowly. So this kind of reflex arc is super, super fast because it's just one synapse, right? Monosynaptic reflex. So you typically can respond to that very, very quickly. Okay. All right. So that interneuron is the connection there in the spinal cord, which is taking information from one nerve and sending that back out to another. And so the organization of those interneurons can look quite differently. We get these different neuronal pools or neural circuits, right, or groups of interneurons. And so just showing you how these different things, we can have some sort of sensation loop with reverberation. We can have multiple sensory neurons feeding into one interneuron, which is all going to stimulate either one motor response or another. We have divergence, which would be one signal stimulating multiple motor responses, right? And parallel processing, which would have multiple interneurons helping stimulate multiple responses to the same signal, etc. So I guess basically some more terminology. Great. So again, monosynaptic reflex will be the fastest and simplest. And our most common monosynaptic reflex would be something like our stretch reflex, or at least one that everyone likely has experienced or seen happen in like a regular checkup at the doctor's office, this stretch reflex, right, where they hit this patellar ligaments with that little tiny hammer, and your leg is supposed to kick out in response. That is from your stretch reflex. So when the patellar ligament experiences stretch, very, very quickly, right, that very quick stretch to that ligament, that is a signal that could potentially lead to damage if your muscles stretch too far or your ligaments stretch too far and pull those fibers apart. That is going to cause some extensive damage that is difficult to repair. And so in order to prevent that stretching from continuing, the receptor sends that sensation to the spinal cord. The spinal cord sends that motor response back out to the effector, and we immediately contract our quadriceps muscle group to extend our knee and try to basically do the opposite movement that we were experiencing. So to remove that flexion, which would continue to stretch this muscle, we extend the knee to try to protect it and prevent it from overstretching. So we only had one muscle or muscle group responding to that one direction of movement in response to that. So very simple, very fast. That is another monosynaptic reflex. Now, we do have a few other reflexes or other types of reflexes that involve a bit more interconnectivity. So from what we've looked at so far, we just have an effector being one muscle or muscle group. So the same action as a response. Okay? Up here, all we needed to do was flex our elbow to get away from that stimulus. Here, all we needed to do was extend our knee to try to prevent that damage. But for some things like experiencing pain in this case, we need to flex and inhibit the opposite side. So rather than just stimulating one movement, we are also simultaneously inhibiting the opposite movement. Because as you reach for this pan, you're basically sending signals from your brain through these motor neurons to your triceps and other muscles that are involved in extending your elbow, telling your arm to extend to grab onto this pan. So while you are still sending those signals, that stimulus is going to cause a pain response in your hand that travels to the spinal cord and is going to get back to those muscles before your brain becomes involved. And so you also need to inhibit that response that you were cognitively sending. So stop yourself from continuously extending your arm and only respond to that reflex that wants you to flex and get away. Okay? So in that case, we have a lot more going on. You're basically fighting what your brain was telling you to do before your brain knows that you were experiencing pain in this reflex. Okay? So that is our withdrawal reflex. It has a lot more going on. We have multiple synapses. These are polysynaptic, whereas these were monosynaptic. It's polysynaptic. So the heat from the pan stimulates your contraction of your flexors. It's also inhibiting your extensors, and it is sending that signal that you experience pain to the brain. So multiple different directions for the signal to go, or multiple different effects from that signal. So similarly, I guess kind of taking that idea and now essentially doubling its effect, because we're not just looking at one limb, but we're going to have two limbs responding. So this is our crossed extensor reflex. So the same basic idea, we experienced pain while we were sending out some other signal. So we were extending this leg so that we could walk. We stepped on something painful. So we now need to counteract the signal that our brain sent, which was to walk. So we need to inhibit the extensors and to stimulate the flexors. But if we just do it to this one leg, we're going to fall over because we were already telling our other leg that we were making a step. So we not only need to have that inhibition of extension and the stimulation of flexion in this one leg, we need to do the opposite basically in the opposite leg to get us back standing so that we don't just immediately fall over. So whereas the leg that experienced the damage is going to have this same response that we saw with the arm. So our extensors are inhibited, our flexors are stimulated. The opposite leg is getting the opposite reaction. It is going to have its extensors stimulated and its flexors inhibited so that you can stand up straight on that leg. So fighting our brain signals in both of our limbs in this case, because we were telling, this is our left leg, I guess. So we were telling the left leg to flex because we were extending our right leg as we were walking, but we experienced pain or experienced the stimulus, and our natural reflex told our legs to do the exact opposite so that we can stay standing up. So this is a crossed extensor reflex. So the sensation came from the right leg, but we're going to have responses in both legs that had to cross across the body. Again, those are all basically signals that are being processed in the spinal cord. So they did not need to involve the brain. They're basically automatic. You can have cognitive control or fight against those things. You can keep your hand on a hot plate even though your reflexes are telling you to get out of there. Okay? Or keep your hand against something. You can keep experiencing pain even though the reflexes are telling you to stop. Okay? But you are naturally evolved to respond in these types of ways to get away from that pain, prevent the damage, do the thing that is biologically inexpensive, more rational. So we brought up the stretch reflex of the patellar ligament as a really common way of testing if these signals are working correctly. Another common test is the Babinski reflex. So naturally, for most of your life, feeling some sort of sensation on the bottom of your foot would cause you to do plantar flexion to try to basically make room for whatever that painful stimulus is or whatever you were sensing there to get away from it. You do plantar flexion to get away from it. But in babies, or essentially for the first 2 years of the average human's life, that reflex isn't set up that way yet. And so rather than plantar flexion, they will actually just spread their toes in response. And like they're kind of just being tickled, like, ooh, spread their toes. And so this is another common check if your neural signals, your reflexes, your reflex arcs are working correctly, because you can show this same Babinski reflex, spreading of the toes instead of that plantar flexion, if you have that neural damage or inhibition to your reflex arc. Okay. And the very last thing to talk about here, muscles that are involved in these reflex arcs have their own terminology to refer to them. So in a lot of our muscles, you'll have these intrafusal fibers. So the extrafusal fibers are going to react normally the way that we've like talked about all of our other muscle cells, or most of what we've been looking at with skeletal muscle tissue. We're looking at extrafusal fibers. The separation here is that intrafusal fibers specifically respond to these reflexes. They can recruit the extrafusal fibers. They can recruit the rest of that organ, the rest of those muscle cells. But we do have a smaller concentrated grouping of these fibers that specifically respond to these reflex nerves. And so those reflex nerves are gamma motor neurons, gamma efferents, which are their axons. So the intrafusal fibers respond to the signals from the gamma efferents, which are the axons of the gamma motor neurons. So these are the muscle cells responding to these reflex arcs. Okay. Those are all of the important things from chapter 13. I guess it's subjective to put it in that way. Those are all the things that you could be assessed on in chapter 13. So we still have a good amount of time. Does anyone have questions or topics you would like me to go back into, or applications of these ideas that we could try to explore and talk about? Um, can I get a quick fist to 5? Asking, I recognize it's a lot of terminology and stuff, and I don't expect you guys to be masters of terminology from sitting through one hour lecture. But I want you to rate your confidence in recognizing like the patterns that we've talked about as far as where sensory information is traveling, where motor information is traveling, and then the stuff about reflex arc. So how confident are you in those conceptual topics? So how information is traveling through your spinal cord and how we respond to things like reflexes. Okay. So mostly twos. So that tells me that there's more that could be said, right? To try to talk about this. Like trying to think of more ways that like I can do better to lead us towards where things are confusing. But I do feel quite reliant on you guys pointing me towards what parts are the most confusing. So even if you don't have questions or ideas that I could try to go back over or say in a different way, discuss. Lisa, are you talking to me? Because you're muted if you are.
I just took it myself. I said more time to study will probably get us there. So that's what I said to myself.
Okay.
I think I really benefited from lab yesterday when you compared like the order of structure to our muscles chapter. I think like a lot of the way that I'm processing this information is already from like those foundational organizational units that you pointed out yesterday in lab.
Um, so for some reason the heat is really loud in here, so I'm having trouble hearing you guys. But it sounded like you were saying you found it helpful that we can connect this structural organization to what we learned about with muscle fibers. Was there a
Yes,
question in
that
there?
was right.
Okay.
Could you hear me? Yeah.
Yeah, I agree. That is nice as a transition here, seeing that same structural organization of the nerves. Any any direction you guys can point me? Okay. Um, okay. Well, if you do have questions, please feel free to send them to me and I can try to prepare responses or expansions and applications for the start of our next lecture. Um, and of course, you're always welcome to send me questions or come to office hours for help at any time. Um, Let me take a quick look at our schedule. Yeah, so we only have 2 more weeks of like covering content of teaching and learning stuff together before finals week. So next week is going to be focused on chapter 14. The following week, we'll address sensory path, more about the sensory motor pathways, and then a bit about the autonomic nervous system, so through chapters 15 and 16. Um, and then 3 weeks from now, it's finals week. So we just have our exam 3 on a Tuesday in lab. So if you haven't already planned around that, please make sure you check out the course schedule and make sure you are available on Tuesday the 19th at 1 to 3 p.m. And yeah, so I guess that's it from me, guys. I hope you have a great weekend, and I will see you next Monday.
Okay. Just quick